Hello, thank you for everyone for joining us today. My name is Lorraine Clark. I'm a journals development editor here at ASM, and the journal M Systems, which is hosting today's webinar, is one of the journals in my portfolio. This webinar will focus on critical concepts in dormancy and is the first one of a two-part series focused on microbial dormancy. In this session, we will cover dormancy in human health and the environment. We're going to begin with a brief overview of M Systems, then we will introduce the speakers and the webinar format before launching into the presentations, which will be followed by panel discussion and Q&A. You are welcome to contribute any questions you may have by entering them into the Q&A section at the bottom of your Zoom panel, and we will try to get to as many of them as possible during the panel discussion. Next slide, please. The journal M Systems is an open access journal led by editor-in-chief Dr. Jack Gilbert. M Systems publishes research in systems biology ranging from an individual cell through entire microbial communities, including the development of new computational approaches to derive biological insights from these large data sets. Next slide, please. Next, I'd like to mention a new M Systems initiative. You can scan the QR code for further details, and we will also provide relevant links in the chat. Aligned with today's webinar topic, M Systems opened a call for papers just last week to our new special collection on microbial dormancy, which is led by Senior Editor Dr. Ashley Shade, who is also the moderator for today's webinar. This collection will feature innovative investigations into dormancy across the topics published in M Systems, including ecology and evolution, metabolism and cellular processes, and antimicrobial persistence and stress responses. The journal welcomes original research articles, methods and protocols, opinions, hypotheses, or prospective articles for this series. The editorial announcing this initiative provides more information, and you can access it via the QR code and a link we will share in the chat. Next slide, please. And now I would like to introduce our moderator for today's webinar, Dr. Ashley Shade, who is a director of research at the University of Lyon in France. Her research aims to understand, predict, and manage microbial communities to achieve resilience to climate change and other environmental disturbances. Ashley joined M Systems as an editor in 2015 and then became a senior editor in 2022. Ashley, over to you.
Thank you, Lorraine, and welcome everyone to this thinking series. This series has been in the planning for almost a year, and so I am so happy to join you here today for part one, which is called Dormancy in Human Health and the Environment. I'd like to start off by thanking our esteemed panelists for generously sharing their expertise today, the ASM staff for their exceptional support in organizing these webinars, and also to all of you for taking time out of your busy day to geek out about dormancy with us. So many cells can persist in a reversible state of reduced metabolic activity called dormancy. Dormancy allows cells to survive unfavorable conditions, disperse and immigrate widely, broaden community diversity, extend lifespan, and preserve genetic information for future generations. Clearly, understanding the consequences of dormancy has wide implications across biological systems and the potential to contribute to addressing grand challenges facing humanity, all the way from controlling disease to facing the climate crisis. So the objective of part one of this thinking series is to bring together international experts who think about dormancy from the perspectives of human health or environmental science, and to synthesize the commonalities and differences that these diverse perspectives offer. So for our first speaker, we welcome Professor J.T. Lennon, an ecologist and evolutionary biologist from Indiana University, the United States. He invigorated research into microbial dormancy with seminal research and a now classic Nature Reviews Microbiology article in 2010. And since then, he has led microbial ecology and evolution in both experimental and conceptual advances in understanding dormancy. Jay, thank you so much for being here, and the floor is yours.
Okay. Okay. Hope you can all hear and see me. Thank you for the opportunity to be here and to share some ideas in this M Systems Thinking series, two-part series. We're going to hear from a lot of interesting people today with very diverse backgrounds, some focusing on environmental systems, some focusing on host systems. We're even going to hear today from people who are applying ideas of dormancy to cancer biology and metastasis. So in an effort to kind of make sure that we're all on the same page, I'd like to provide a gentle introduction or background to some of the fundamentals, at least from my perspective, on how we can all be thinking about dormancy. And I'm going to start with a working definition, and we can discuss this. There might be some people who want to add components or disagree with it, but it's a process that operates upon individuals. And I think that's an important thing. We're going to start thinking about individual cells. It could be multicellular organisms or any kind of agent that interacts with other agents and has the capacity to reproduce. And so those individuals have to enter a reversible state of reduced metabolic activity. And so what I'm going to do right now is just walk through 3 requirements or criteria that need to be fulfilled in order for an individual to engage in dormancy. So first, that individual has to be able to occupy different metabolic states. And for simplicity, I'm just showing 2 potential states that an individual could occupy, either on or off, active or inactive. In reality, an organism may be able to occupy metabolic states that exist along a spectrum or continuum of metabolic activity. We know that there are certain developmental programs that have distinct stages that need to be passaged through in order for a cell or an organism to become inactive. But again, for simplicity, we can just think right now of there being these 2 metabolic states. So that's criterion 1. The second criteria is that an individual needs to be able to transition between those 2 states that we're considering. And those transitions can happen in one of 2 qualitatively very different ways. So an individual may experience environmental conditions that are not suitable for growth and reproduction. And they may be able to sense those conditions. They may have receptors on the surface of their cells. They may have certain machinery that allows them to detect changes, for example, in temperature, pH, the availability of resources. And when those conditions become poor, the individual can sense that and they can respond accordingly and move into an inactive state. Another important feature is that for dormancy to be evolutionarily advantageous, you can't just stay in an inactive state forever. There's no distinction between that and being dead. So organisms need to be able to sense, in some cases, when the environment changes and respond accordingly. And that's a process known as resuscitation. So what I've just described are conditions where there are responsive deterministic switching, where cells are making decisions based on conditions in their environment. But in some environments, the conditions change so rapidly, so unpredictably, maybe even chaotically, that the investment in the cellular machinery to detect and respond to those changes is not optimal. And so in some environments, and I think we'll hear about this a little bit later, is that cells can switch or individuals can switch between metabolic states in a stochastic or random way. And that fulfills what some people refer to as a bet hedging strategy. Okay, so that's criteria 2, the transitions, those arrows that represent movement between states. The third is that there are some demographic processes that we need to consider. So in the red circle, the active individual, you can see that there's this arrow that returns back upon itself, and that represents growth of an active individual in a good environment. So that's the advantage in a stable, good environment, is that you can reproduce and make copies of your genome. When the conditions are bad, however, those individuals are going to experience greater mortality. And so when you become inactive, you're not able to reproduce, but you enjoy protection or a reduced rate of mortality. And that is the third essential criteria, is that when you transition from an active state into an inactive state, there's a trade-off in that you cannot grow and reproduce, but you have higher survivorship. So those 3 things must be met, at least from my perspective, in order for us to be thinking about something as being dormant. And everything I've been talking about so far has been operating on the scale of an individual. But there are interesting properties that can emerge from dormancy when we start to think about the collection, the aggregation, the sum of all of the individual cells, if you will, in a population or a community. And that's represented in this seed bank model that I'm showing on the side here. And each of those colors corresponds to different individuals, and those different individuals have different types of information. Those individuals could belong to a population. They may harbor different mutations that perhaps allow them to persist certain drug treatments. They may be more or less susceptible to virus infection. They may contain certain genes that allow them to assimilate novel substrates. All that information goes into both the active pool of individuals and the inactive pool of individuals. So why is dormancy beneficial, and what does the seed bank do? One of the things that it does is that it buffers populations against extinction. And that could be good or bad depending on your perspective and why you care about dormancy. But it buffers against extinction and allows for persistence by averaging out population sizes over fluctuating environments. As Ashley pointed out, another kind of underappreciated property of dormancy in seed banks is that passively moving individuals, like small cells that may be picked up on currents, for example, or flows, can be moved between environments, and the probability of them surviving in transit can be increased if cells are robust to environmental conditions that would otherwise cause them to die. So dormancy can facilitate dispersal and therefore has an important component not only on time, but also on spatial aspects of microbial dynamics. And together, the storage of information in the seed bank provides a memory, which means that things that happened now or in the future are going to depend on information in the past if there are dormant individuals that are resuscitating. So this creates structure in populations or communities. It operates across vast spatial and temporal scales, across levels of biological organization. We can think about the evolution of individual strains or populations, or we can think about the way in which seed banks can confer and promote stability of whole ecosystems and the biogeochemical processes that they regulate. I want to briefly talk a little bit about the origins of dormancy, because I think it's important for how we design experiments, how we develop assays, and ultimately how we study dormancy in complex microbial systems. So I'm depicting a phylogenetic tree here, and there are three distinct monophyletic clades represented by different colors. And these could all be different flavors of dormancy. And I point this out because whether or not we're thinking about microbes, plants, or animals, dormancy is an example of what people call convergent evolution. Dormancy has arisen independently multiple times through Earth's history in the tree of life. What that means is that there's no common way in which organisms engage or carry out dormancy. And that should be important for people like microbiologists who think about using genetic tools for characterizing certain traits. It also is kind of interesting from a biological perspective when we see convergent evolution, because it means that life has somehow figured out a solution to a universal problem. In the case of dormancy, it means that organisms have figured out a way to deal with living in fluctuating and unpredictable and harsh environmental conditions. At least among microorganisms, there are some interesting evolutionary aspects related to what I've called use it or lose it. Some forms of dormancy are made up of complex genetic regulatory programs. And if those organisms are living in good environments for prolonged periods of time, there can be mutations that will accumulate randomly in the genome and will disrupt certain pathways that are involved in dormancy, meaning that even traits that have been around for millions or billions of years can be lost very rapidly. And in the process of that, what we found is that even if you lose one form of dormancy, there are certain microorganisms or lineages that have secondary or backup versions of dormancy. So not only do we need to think about there being monochromatic lineages here, we need to think about that there's multiple overlapping traits and backup mechanisms of dealing with survivorship via dormancy. And like many of you know, many microbial traits compared to plant and animal systems have the ability to not only inherit information vertically, but to transmit that horizontally. And so we also need to think about, in certain cases, how traits that are encoded for dormancy can move among distantly related lineages. And so hopefully you can see why those things might be important for how we study things, at least operationally. I'm going to talk a little bit briefly about the diversity of microbial systems and it pertains to the mechanisms and phenotypes across different lineages. So trying to kind of embrace this idea that there are different flavors of dormancy. And microbiologists have spent a lot of time understanding development and morphological characteristics that are easily identifiable under microscopes. On the left-hand side, I'm showing a picture from our laboratory where we took some images using phase contrast of endospore-forming bacteria. So strains or lineages like Bacillus and Clostridia, some of which can be used for probiotics, other ones that cause certain diseases that are difficult sometimes to treat with drugs, and in some cases, strains that can even be weaponized by using spores. So for example, in Bacillus anthracis. In this particular example, endospore formation is a very complex developmental program. It involves hundreds of genes, scores of proteins, a lot of time and a lot of energy. It's a very complex trait, and it's been around on Earth for about 3 billion years. We also see examples in the middle of myxospores by Myxococcus, exospores, which are different from these other versions of dormancy that are produced by certain Streptomyces strains. And on the right, my only eukaryotic image is that of mycospores. So these are spores that are produced by arbuscular mycorrhizal fungi, which I think we'll hear a little bit about later. And these fungi can aid in the resource acquisition for the plants and form these symbiotic associations with about 90% of all flowering plants or angiosperms. So these are some very conspicuous forms of dormancy. They're often very complicated forms of dormancy, and in many cases, they've been around for a very long time. I think one of the things that we need to acknowledge moving forward in this series is that there are many less conspicuous forms of dormancy, some which are well described, all of which are probably a little bit harder to study, and probably a whole bunch of different types of dormancy for which we've yet to discover. Some of these include things like resuscitation promoting factors, Mycobacterium tuberculosis, the causative agent of TB, has these resuscitation promoting factors which allow them to wake up. Important and relevant for human health because about 1/3 of the world's population is walking around with latent or dormant infections of Mycobacterium tuberculosis. Certain bacteria, both in host systems and in the environment, engage in a phenomenon known as viable but non-culturable states. There are proteins that can modify ribosomes, which are important for protein synthesis. And there are persister cells. There are various ways in which persister cells can engage in dormancy. But in the simplest scenario, you can have a single operon with 2 genes that can control the way in which cells make decisions to enter and exit from dormancy. So I'll leave you with a question and maybe a direction that we can consider, that there may be some universal measures of dormancy, fundamental ways in which we can think about the physiology and the metabolism of the cell, which is core and central to thinking about dormancy. So one of the papers that we worked on very early when I first started thinking about dormancy was looking at the ribosome content of single cells. And this was done with fluorescent in situ hybridization at the time, and none of these data were generated by us in the lab. This is information that we synthesized from the literature. And so we could look at the number of ribosomes in cells as a proxy or measure for inactive cells, or the size of a seed bank, if you will. And you can see across different ecosystems, guts, wastewater treatment plants, oceans and freshwater and soil environments, that there's a lot of variation among ecosystems. And in some cases, soils can have 90% of all the individuals in a sample being metabolically inactive. Other ways that we could think about classifying or characterizing dormancy in microbial systems may leverage things like the abundance of ATP molecules inside of a cell, the degree to which membranes are energized, the activity of the electron transport chain, We could think about ratios of nucleic acids. So RNA is an important component, obviously, for protein synthesis, but it also makes up a large fraction of the ribosome, whereas DNA is a relatively static and low concentration ratio. So if we look at the ratio of RNA to DNA, that could serve as a proxy for dormancy or the relative activity of cells. There are also new methods that are being used to measure rates of protein synthesis at the single cell level. So I don't have the answer for this, but I think that some of these types of measurements will span the variation, the diversity, the flavor, and the morphological characteristics that may vary among lineages of microorganisms that engage in this common strategy of dormancy. And I'll just leave you with one thought, and that is to ponder how dormant is the microbial biosphere at a global scale. We know that there are many, many species that are persisting and maintaining diversity over vast timescales when challenged by environmental conditions. It's likely that dormancy is one feature that contributes to the maintenance of that diversity. There was a study a few years ago that attempted to quantify the number of endospores, again, which is just one way in which one lineage of microbes can carry out dormancy, and estimated in marine sediments alone that there are 10 to the power 28 endospores on the planet. Independently, we have pretty constrained estimates on how many microorganisms there are on a global scale, and that number hovers around 10 to the power 29. So conservatively, a very back of the envelope calculation is that about 10% of the global biosphere is made up of a seed bank, and that only considers one of these flavors in one environment. So we can start to think about perhaps how all of our individual studies and all our methods may contribute to refining those numbers and thinking about the way in which dormancy influences the activity and diversity of the world we live in. So I'd like to just thank you all for attending and your attention, and I'm really looking forward to discussion that follows in this session and the other thinking session that will be in November. Thank you.
Great. Thank you so much, Jay, for that great first talk. Reminder, if you have questions for Jay, you can put them in the Q&A section, and we'll get around to them at the end of the talks during the panel discussion. Okay, so for our second speaker, we welcome the esteemed Professor Natalie Q. Balaban, a physicist at Hebrew University of Jerusalem from Israel. She is known for her contribution to defining and understanding the dynamics of microbial persister cells that evade antibiotic treatment through phenotypic heterogeneity. She has also pioneered the use of microfluidics and automation to quantify single-cell responses to antibiotics and their consequences for antibiotic tolerance and resistance. So Natalie, thank you so much for joining us, and the floor is yours.
Yeah, it's a pleasure to be here. And thank you, Jay, for the great introduction to dormancy. So what I'm going to talk about is falls in the realm of these persister cells, namely cells that are growth arrested. This is what we see. And because they do not grow, they are also more protected against many antibiotics. And what I'm going to talk about is the fact that there are many ways to be growth arrested, and not all of them would qualify to the dormancy definition that Jay has introduced. So what we see is that when we have a dormant state, and the endospores, for example, are a very good model for it, and here this is a movie of endospores forming, what we see is actually a very regulated program with specific genetic pathway that leads to a growth-arrested cellular state, which qualifies as a dormant state, and what we call a regulated growth arrest. And this will come in opposition to something that is non-regulated, which is actually a chaotic growth arrest. But this is very clearly a regulated growth arrest. It has evolved to protect the cell against many stresses. Now, if we work in non-sporulating bacteria, such as E. coli, we also have regulated growth arrest. For example, when we starve bacteria, E. coli, for amino acids in minimal medium, eventually here you see the growth curve. So the cells will grow exponentially, and then they will arrest their growth, and the optical density here stays constant for several days. And also the viability stays constant. So E. coli can be growth arrested and maintain this growth arrest mode for extremely long period of time, not as much as spores, of course. And what we see is that they do have some activity in this period, but this activity is much lower than exponential growth. So they have an activity during exponential growth, and here by protein production, we can measure this activity. And upon the entry into this stationary phase, this growth arrest, the activity goes down very dramatically, and now we have an activity that maintains this constant level of activity for many days. And this is what we call the Cas state, which is a very regulated growth arrested state. And here we can flow the culture of these growth arrested cells into the microfluidics and see how the E. coli enter this growth arrested mode. So here they grow and then they are growth arrested. And in order to enter this mode, you can see there is a reductive growth. So they form a smaller bacteria, and then they can stay like that for many days without growing, maintaining a very, very low level of activity, but still measurable activity, so that they do produce protein if you ask them to. So this constant activity stationary phase, as we call it, is a form of regulated growth arrest. So it is dormant compared to actively growing cells, and it is also reversible so that they can regrow from this state. So what we picture this growth arrest mode is a very organized, so the cells were cycling, and now they're, because conditions change, they are parking their bike in a very orderly fashion, and clearly this regulated growth arrest it depends on many pathways, such as the starvation response and the strain response. Now, what do we call disrupted growth arrest? So if we take this biking analogy, the disrupted growth arrest would be what happened in the Tour de France a few years ago, when the peloton was cycling, and then one of the fans on the side waved a sign, and this is what happened, right? So as you can see here, the bikers stopped biking. They arrested the cycling, but in a very disrupted mode, right? And if we asked how can we do that for bacteria, actually we do it regularly when we expose bacteria to, for example, bacteriostatic drugs. So here we are growing E. coli in this microfluidic device, but now we are not going to starve them naturally. We are going to starve them acutely and very unnaturally. So they freeze. I don't know if you see that, but they completely froze their growth. And they do not produce these smaller bacteria, so no reductive growth. And when we wash away the acute stress, they restart growing, but some of them, as you can see here, for example, in the middle, some of them start growing immediately, but some of them will start growing only half a day after the stress has been taken out. Okay, and that's one of the signs of what we call a disrupted growth arrest. So this acute stress leads to this disrupted growth arrest, no reductive growth, and very slow and variable recovery from the growth arrest. And when we quantify this recovery from the growth arrest, so these curves here show how the growth arrest is how bacteria return to cycling again. So at the beginning, when we take away the stress from the disrupted growth arrest, bacteria do not grow. And as they grow, the number of growth arrested bacteria go down. But the longer you keep the stress on, and the different colors here are different lengths of maintaining the stress on the bacteria, the more distributed and broad is the recovery time from this growth arrested state. So some bacteria, after an hour of, or 2 hours or 3 hours of stress, will recover within 30 minutes. Others will take 1 or even 2 days to recover. So The disrupted state is actually an archetype of non-growing growth arrest. And it's characterized by this very highly variable recovery from growth arrest back to growth. And this growth arrest and this recovery depends very, very strongly, as you see in the colors here, on the duration of the stress that the bacteria were exposed to. So the duration of the growth arrest is going to influence how fast these bacteria or how slow these bacteria are going to recover. If we go back to the regulated growth arrest that we saw before, now if we do the same experiment on the regulated growth arrest and ask how fast do bacteria recover from a regulated growth arrest, so now you can see that it's dramatically different. The recovery from a regulated growth arrest is very fast. It happens within 40 minutes. And what is striking is that it does not depend at all on the duration of the previous growth arrest. So E. coli may have been halted in this regulated growth arrest for a day, 2 days, or 3 days. When we replenish a nutrient, they immediately start growing, and they are not viable at the single cell level. So these are the two archetypes of non-growing bacteria, and we call them non-growing persisters. Why? Because the very fact that they are non-growing equally protects them from various antibiotic treatment. For example, here we see two culture, one kept for 2 days at the regulated growth arrest and one kept for 2 days at the disrupted growth arrest. And as you can see, when we add ampicillin to the arrested culture, they are equally protected from ampicillin treatment. And the same treatment would have killed 3 orders of magnitude of exponentially growing cells. So the disrupted and regulated growth arrested bacteria are both persistent to this beta-lactam treatment. Now, what is interesting is that if you look at persistence pathways, for 20 years, people have been working very hard on finding pathways for persistent, which should be analogous, if we thought about it, to, for example, to sporulation pathway, but no clear pathway has been identified in 20 years of research. And our explanation is that because most of the persisters that we study in antibiotic research are actually disrupted growth arrested bacteria. And because this disrupted growth arrest is very chaotic, actually, it's not driven by a specific pathway as for the regulated growth arrest, but it's actually driven by chaos, and therefore there is no hope of finding a pathway for this growth arrest. Now, what can we say about this very disrupted growth arrest? So what is nice about this disrupted growth arrest, that because it's chaotic, actually we have a model for it that is taken from biophysics. I'm going to enter into it, but this model actually completely predicts these recovery dynamics, the fact that they are very variable and very depending on the duration of the growth arrest. And second, the model predicts that not only in the recovery, also in, for example, if you try to measure protein production, there should be a very high cell-to-cell viability in the disrupted growth arrest as we measure. So what can we say about these disrupted bacteria? The regulated one activated pathways that are very well known of taking down, for example, transcription very much upon the growth arrest. So what we see here, if we measure the total number of mRNA per cell in the exponential phase and in the regulated growth arrest, you see orders of magnitude decrease in mRNA transcription in the regulated growth arrest, and that makes sense. If we want to be dormant, we don't need to produce all these proteins, and therefore we do not activate transcription, and this is the regulated growth arrest. In contrast, the disrupted growth arrest doesn't reduce its mRNA level despite the fact that it's not growing anymore. And this is the first hint that these cells are actually completely defective in mounting a proper stress response in order to enter dormancy. So they are not dormant, okay? And the first hint that they are not dormant is the fact that they do not reduce the level of the mRNA when they are growth arrested, in contrast to the regulated one. Another hint is in the transcriptome is that we see that the disrupted growth-arrested culture do take down the flagellar genes and the ribosomal genes, but not as much as the regulated bacteria, and therefore they are still maintaining this high level of activity. In the physiology, as we see, there is no reductive growth in the disrupted bacteria, so this is another hallmark of the disrupted state. And finally, of course, if we maintain for more than a few days the growth arrest of the disrupted bacteria, they will eventually die. And this is, of course, another hint that they are not entering a proper growth arrest. Now, another hint to the fact that despite the fact that they are not growing, they are not dormant, is by the measurement of the heat that the culture at disrupted state produces. So that's a very neat measurement, because it's really completely oblivious to many details. You just take the culture in the disrupted state, in the exponential state, or in the regulated growth arrest, and measure the total amount of heat that is produced by this culture. So if we measure that by microcalorimetry in exponential phase, we see that exponentially growing cells are producing about half a picowatt per cell, right? That's more or less what is measured. In many different bacterial species, this number is pretty much conserved. Now, if we starve the cell and let them enter this regulated growth arrest, the CASP state, as we see, the heat production per cell goes down very much. And this is the dormancy, right? We do expect a very clear metabolic reduction in dormant cells, but in the disrupted growth arrest, we see that the metabolism is actually not going down at all compared to exponentially growing cells. So the failure to shut down the metabolism, the failure to mount a proper stress response is what characterizes this disrupted bacteria. And therefore, because they fail in doing so many things, maybe we can kill them differently. So in the persister field, because persister bacteria are non-growing, it was suggested that you can supplement them with nutrients, make them grow, and then be able to kill them. So this strategy works very well on the regulated bacteria, the one that are maintaining their dormancy very tightly. Upon nutrient addition, they all start growing very quickly, and therefore, indeed, if you make them grow quickly, you can kill them. But it doesn't work, and the disrupted state are actually not affected by adding nutrients, because they have this very broad and slow recovery from the previous stress. So, but understanding the fact that they are not in a regulated state, they've crashed and they are trying to maintain their homeostasis, gives us hint of how we can actually kill them. And one first hint is the fact that the membrane homeostasis is disrupted, as expected from the absence of regulation of dormancy. They are permeable to PI, and therefore they are killed by antibiotics that are not killing the regulated persisters. So here we see, for example, by the adhesion of gentamicin, the gentamicin is not killing the regulated dormant persisters, but is killing very effectively the disrupted ones, and understanding this disrupted state leads to other targeting compounds as well. So I think I'll skip this part and go to the summary. So what we've seen is that it's not enough to arrest the growth of bacteria to really make them enter a dormant state that is protected against many things. Unnatural starvation, abrupt stresses, all kinds of bacteriostatic antibiotics actually lead to a disrupted growth arrest. And this disrupted growth arrest can be modeled by a biophysical model that actually predicts how they are going to behave. They fail to mount a stress response and result in a loss of homeostasis. They are highly variable. Cell-to-cell variability in disrupted growth arrest is very high, while it's very low in the regulated one. And despite the fact that both regulated and disrupted bacteria are highly persistent to antibiotic treatment, such as ampicillin, as we saw, that target growth, we can actually kill the disrupted bacteria by using a compound that alter the homeostasis of these disrupted bacteria even further and helps us to get rid of them. And finally, I'll say thank you to all the people that did the work, mainly the work of Yoav Kaplan here, who did the first work on the disrupted state, and Adi Rotem, who continued on this variability, a single cell variability in the disrupted state. Our collaborator, Oder de Gam, and funding from the ERC. And thank you very much for your attention.
Great. Thank you so much, Natalie. That was a really interesting insight into how we think about defining dormancy and what the characteristics of dormancy are. And it really springboarded from Jay's discussion as well. So I hope we get around to that in the panel discussion. I'd like to, if you have questions for Natalie, please add them to the Q&A. And I'm going to now introduce our third speaker. So we welcome Professor Pilar Junier, a microbiologist at the University of Neuchâtel, Switzerland. She is known for her research to define, quantify, and understand a diversity of dormant phenotypes from environmental microorganisms, with a special emphasis on spore formers. Her current research also extends to ask how dormancy and related adaptations can be leveraged to develop practical biotechnological solutions, thus offering an applied sciences perspective to our discussion. So thank you very much for contributing your perspectives, Pilar, and the floor is yours.
Thank you. I'm going to start sharing. Just please let me know that it looks good on your side. Okay. So first of all, I wanted to thank again Ashley to put this panel together. I'm really, really happy to be able to present a bit of the work we have been doing in Neuchâtel, and also to be able to discuss later on with other panelists. I think it's very interesting. And I will go a bit back to what Jay was presenting at the beginning. And we have been focusing one of these types of structures that are quite identifiable for dormancy, which are spores. And the question for us was at the beginning, okay, why will a spore or a spore former decide or not to sporulate? And initially we were focusing on bacteria, but I hope that towards the end of this talk, I will convince you that we need to expand a bit our definitions and also our concepts and consider rather microbial survival. And I like to start my presentations backwards. I like to start by really thanking the people who had done the job. So these are a number of PhD students that are brilliant, and also a bit advanced scientists that had participated to this research. And for me, working in dormancy is a bit like being in a permanent reading mode. And I think I'm a big fan of Harry Potter, and I think actually working on dormancy is a bit like reading the last book of Harry Potter permanently, because it's a struggle about survival. And I think this is really the key term that we need to remember. And Jay mentioned it, is about how to survive. And obviously there is not one answer to this question. And already in this book, you see there are two strategies, they have trade-offs. And this is really what we need to consider when we are investigating dormancy in the environment. And for me, the very first wake-up call for this considering dormancy was actually a sentence in this book. I'm not sure, I don't know how many of you maybe know about it. Cannot see it. Environmental microbiology, from genomes to geochemistry. And I think what is important is this is one of the core definitions of this book, is that the prime directive of microbial life is survival, maintenance, generation of ATP, and whenever possible, sporadic growth. And for me, that was very surprising, because microbiologists were used to work in conditions that are not at all favoring this or being coherent with this prime directive. So we tend to study microorganisms in the laboratory under conditions that are actually favoring growth, which is not what very likely happens when you start considering microbial communities in real environments. So for me, this dissonancy between what we do in the lab and what we expect to find in the environment was really what drove me to investigate dormancy. And there was already quite a number of studies that showed that this is indeed the case, that we have really a problem between what we do in the lab and what we observe in the environment. And this can be seen by this paper initially in 2004, in which if you look at the diversity of organisms that have been cultured and that exist in culture collections, you will see that there are a few numbers that are really high And those include some of these organisms that Jay presented at the beginning, firmicutes that produce endospores, actinobacteria that produce exospores. So those are very, very common in culture collections, but the very first comparative metabolomic or metagenomic study that was conducted by Christian von Mering in 2007 found something quite striking. He found that actually these organisms that are extremely common in culture collections were somehow absent in environmental samples. And the question that he posed in this paper was, okay, is this a real signal of dormancy not being common in the environment, or rather a methodological bias that we have been ignoring when we prepare, for instance, DNA samples for metagenomics? And this is really where we started as a team. So our goal was to develop approaches that would allow us to look at dormancy in environmental samples, considering two types of combined methods. So on the one hand, we wanted to develop culture-independent approaches that allow us really to look specifically for these organisms in a wide range of environmental samples. And on the other hand, we wanted really to keep on culturing organisms that may have one of these structures of survival, independently of what were their initial phylogenetic affiliation. And obviously today, I will not talk about everything. I will mainly focus on the first element, this methodological bias for the detection of spore formers in environmental samples. And I would like to give you this idea of cryptic sporulating taxa that we need to continue investigating and understanding. So to start with, the first thing that we had as a hunch was that, okay, we think that, and we thought that the reason why we were missing these endospore formers, partially in these metagenomic libraries, was basically that we were not obtaining DNA from these organisms. And that came from a series of experiments that we did in the laboratory with, for instance, endospore formers, in which we saw that most DNA extraction kits, standard DNA extraction kits, failed to obtain DNA. And because of that, what we decided with Tina, who was the first PhD student working on the topic, was to develop a method in which, starting from an environmental sample, we will somehow separate first a fraction of the community that is easy to break. This is the community that appear usually in any metagenomic study, and focus on breaking this easy to break or easy to lyse community first, get rid of that DNA, and keep in the sample whatever was resistant or persistent or lysis resistant, which is what we call them now. And with that method, what you have is then a community that is split into two fractions. One fraction that is, let's say, the total community, in which you have a mix between species that can be lysed easily and species that may be harder to lyse, and a second community in which you really focus only on these hardy structures, which are really what we call, what at the beginning we used to call the seed bank, now we call just the lysis resistant fraction. And then what you can do is then in both fractions, extract the DNA and convert them. And this is something that Tina tested, and she tested this initially in sediment samples. And what she observed is that indeed, when you do this total community, and here are examples of the typical organisms that you will expect to have these lysis-resistant structures like firmicutes, what you can see here is when you do this whole community analysis, your fraction of firmicutes is relatively low. But when you do this endospore or lysis-resistant fraction, your fraction of the community increases dramatically. So that shows us that indeed there was a problem with the way we were extracting DNA from this structure from environmental samples. And she went a bit further and analyzed this in a number of sediment samples that we obtained, in this case from Lake Geneva. And what you can see here is we have samples that were either analyzed with this total DNA extraction or with the lysis-resistant fraction in two colors, in blue and in red. And when you do an ordination analysis to try to compare the community composition, what you see is that there is a clear separation between these two communities along this axis 1. And what is explaining this axis is basically the treatment prior to DNA extraction. So it's clear that when you do these two separate DNA extractions, you get different images of these communities. We went further and tried to see, okay, which groups are explaining this difference. In the case of Lake Geneva, what we observed is, well, it was that firmicutes basically were only or almost only appearing in this lysis-resistant fraction, which means that when you look at the total community, firmicutes will be absent. So clearly these firmicutes is one of these groups that is not rare, but is highly missed in communities when you don't do this specific treatment, pre-treatment for DNA structures. We tried to look at this a bit more into detail. So we took OTUs and we just matched the OTU presence in the lysis-resistant fraction in orange and in the total community in blue. And we compared what was the ratio between the frequency in each one of these communities. And you can see here in this graph that basically almost all the groups that we were able to detect, with exception of this Bacillus OTU here, were only or almost only present in the lysis-resistant fraction. And for me, this gives already a few key messages. First, that firmicutes are present in the environment very likely as endospores. That also you have a number of groups for which we have never seen endospores, and this is what we call these cryptic endospore formers because they appear in the lysis-resistant fraction, and the most plausible explanation for this is that they produce a spore. And it's also a cautionary tale for people working with firmicutes. I've seen a lot of the questions about human health. Actually, in humans, we also think this is an important fraction. But the question is this fraction ever active? Is this fraction ever reproducing? And if you work in biocontrol, for instance, Bacillus is one of the most widely used biocontrol agent. If you use it in soils, will there ever be other than endospores? Will they ever germinate and be active? So it's things that you need to think when you're developing biotechnology with endospore formers. However, we wanted to go a bit further, and what we did was we applied the same methods in a larger range of environmental samples, and this is work that was led by Thomas and Christoph. And what we observe is that depending on the environmental samples, firmicutes, which were until that point the group that we always have seen as dominant, are not dominant everywhere. So in some cases, you start seeing groups like actinobacteria that is here in red, or proteobacteria that is here in gray, as being highly abundant in some specific samples. And initially we thought, Jay presented this at the beginning, we thought, okay, this is normal. You have exospores in actinobacteria and mixospores in proteobacteria. This may be those organisms, but it's not true. We have seen now that there is a number of other potentially sporulating bacteria, and I put this in question mark because it's not easy to demonstrate they sporulate, that are there in the environment and we know very little of. So this is what Andrea tried to investigate during her thesis. She tried to identify chemical markers that were common to all these potential spore formers. So what she did, she used a combination of methods, and today I'm going only to talk about Raman spectroscopy. I don't have the time to show the details of the method, but I'll be happy to explain it later on. But the idea was to try to identify a universal chemical marker. So she did this with these 5 groups that Jay mentioned at the beginning. So mixospores, exospores, endospores, akinets from cyanobacteria, and we include some cyst from proteobacteria. And what she did was she compared the signals, the chemical signals that you obtain when you have the cells in a state that is in reproduction, so vegetative state, or as a dormant state. And what she observed is that indeed, each one of these structures, depending on the state, has a chemical signature. And you can find individual markers of dormancy for each one, or for this modified structure for each one of the groups. However, we wanted a universal marker, and we were very disappointed because we could identify individual markers for each one of these groups, but never something that was common. For instance, we could very clearly identify calcium dipicolinic acid in endospores, which is a marker that is widely used to evaluate endospores. We saw some modifications like tetralols or some other components that are known to be produced during sporulation in these organisms. But at the end, some unknown chemical markers, but at the end, we never found this universal marker. So this is still something we are working on. And now, towards the end of my talk, I told you I wanted to mention that sporulation, whatever you wanted to define it like, is not exclusive to bacteria. And in particular, there is a group that we tend to forget when we talk about microbial ecology, and it's fungi. And why? Because fungi, in the same way as bacteria, they produce a lot of these structures. Some of them are not necessarily involved in survival. They can be involved in dispersal simply, but some are actually important during the life cycle. And what we tend to forget about fungi is their immense diversity in terms of morphology and structures. And this is something that my colleague Saskia Binchadler here and Dana, who is a PhD student, had been investigating for many years in the lab. And in our work, what we did was we also validate these methods that we have been developing for endospore formers in the case of fungi. And we have seen that indeed, if you apply these same methods, these total DNA extractions versus lysis-resistant DNA extractions, you can demonstrate that a large fraction, again, of the fungal community is only present in the environment in the form of a highly resistant structure that we don't know yet if they are dormant or not. So this is really what we will do now in the future. We will start to investigate, can we really demonstrate if they are dormant in the sense of the definitions that Jay mentioned at the very beginning? Can we show specific features? And to do this, we are going to start investigating now, since a few years, we have been working with these systems, deep subsurface geothermal reservoirs. Why? For a particular reason, is that in these systems is the only place or the only systems that we had found so far in the ecosystems we had investigated, in which when you do this total and lysis-resistant fraction, what you see is that there is a full overlap between the communities. You don't see this image that you saw in Geneva with one side, one community, the other side, other community, but a full overlap. And for me, this means that endospore formers are highly abundant in this type of deep geothermal reservoirs. So this is the next frontier for us. And with that, I just wanted to give you a few final take-home messages. First of all, I hope that I convinced you that we also need to think about the methodological biases that we are introducing in our analysis, and also what we need to consider when we are interested in a particular type of organisms, in particular dormant organisms. I think it's important that you consider how to tailor or develop a better DNA extraction method, or at least use a better DNA extraction method if you're working with these type of organisms. I think it's important to try to develop these more universal methods to continue investigating sporulation in the environment and human health. And I hope that you will all, or some of you at least, will move into investigating sporulation not only in bacteria, but also in fungi and archaea, because there are a lot of tons of open questions and a lot of careers that can be done and developed in this area. And with that, I wanted to thank you for your attention.
Great. Thank you so much, Pilar. That was really insightful, and it's interesting to see the differences between the total and the lysis resistant pools, and think about the implications of that for the seed bank. I'm going to introduce our final speaker. If you have questions for Pilar, please put them in the Q&A. We welcome Professor Joel Brown from the Moffitt Cancer Center, an emeritus distinguished professor at the University of Illinois, Chicago, United States. He is directing his deep expertise in ecology and evolution to the understanding of cancer. His latest work is to model dynamics of dormancy for tumor cells as ecosystems, and to ask how those dynamics can be leveraged to inform more effective treatments. In his previous research, he studied seed dormancy in plants, and so he notably adds a eukaryotic perspective to our discussion today. Joel, thank you so much for taking part in this webinar, and the floor is yours.
Oh, thank you so much, Ashley. It's really a pleasure to participate in this. And in particular, I would like to draw attention to the amazing presentations. I'm in awe of Jay and Natalie and Pilar for those amazing talks. And it's a pleasure to be able to share with you where cancer might fit into this kind of picture. So first, what I want to do is invite you to have a more expanded view of what cancer is. So these are the standard definitions that you will see just about anywhere. And in particular, it's generally described as cells that have gone wrong, uncontrolled division, abnormal cells. They might be able to invade other parts of the body. What I'd like you to invite you to kind of expand your perspective and think of cancer as a speciation event. It's really a jumping levels of selection. Imagine a cell of our body, of a whole organism, part of a whole organism trait, actually jumps levels of selection and becomes its own new, in this case, I guess, eukaryotic protist or microbe. What makes it unique is it's jumping from a multicellular organism to being a single-celled organism. Furthermore, unlike standard microbes or microbial infectious diseases, its host is its entire planet Earth, except for the very, very rare circumstances of communicable cancers. They die with their host. So the three things I'd like to do is revisit, which I thought was Jay's really excellent introduction to dormancy. So kind of dormancy as an adaptation, and then evaluate whether cancer satisfies or do we see dormancy. And in fact, I'd like to show you that, yes, there is a particular persister state that was described very, very nicely by Natalie, in which it becomes a huge problem for therapy resistance and, in fact, for the metastatic disease. So generally, we think of dormant organisms as non-reproductive, little to no feeding, reduced metabolic rate. It represents a resistant life history state. And I think a key, the bottom down there, is some sort of reactivation cost. So in switching back to a more active, proliferative state requires a cost of upregulating things. As an adaptation, we've already seen it's a great way to bet hedge against uncertainty. It has been shown to be a way of avoiding overcrowding. In some organisms, it's been shown as a way of avoiding SIB-SIB competition. It provides a certain kind of habitat selection in time in which you can kind of hunker down during bad times until things get better, and you can avoid adverse conditions. Now, when we start to apply this to cancer, I think what I will probably like to suggest is, first, in cancer, the term dormancy has been around for a long time, but it actually developed quite differently and in parallel and without recognition of dormancy in the rest of nature. Furthermore, I think in terms of the attributes, I'm going to hopefully show you that actually cancer cells probably don't satisfy the rigorous definition of dormancy as presented by my colleagues, but as an adaptation, as a state that serves those purposes that we just described. I think you'll see that cancer does fit dormancy to a T. I have to thank the team that I've been able to work with at the Moffitt Cancer Center in terms of trying to establish an evolutionary ecology of dormancy. Here's kind of the history, a little brief history of the use of the term in the cancer literature. So the earliest we could kind of find, or I could find, and I invite any of my colleagues around the world out there listening to this to suggest earlier citations. But Willis, interestingly, suggested that dormant tumor cells were those that actually disseminated from primary tumors, resulted in the metastases, and represented simply a growth-arrested state, which can mean a lot of things. By 1954, it was seen as a temporary state of mitotic arrest, which in a sense would be in the cell cycle going into G2M in the cell cycle and just stopping. It then later was actually suggested that dormancy is actually a small population of cancer cells that are in fact proliferating, but they're neither expanding or contracting. So in that sense, it's almost a dormant population with respect to population growth. And then finally, there was a suggestion that tumor dormancy, whatever that meant, was strongly influenced by the immune system. And this is known as immunoediting, that at a certain size, the immune system can control and, in fact, even eliminate a population of cancer cells. But if the cancer cells expand beyond a certain size, the immune system is unable to control it. So here's kind of a schematic to be thinking about all of these ways in which dormancy was being viewed in cancer. So one is this kind of small population definition of dormancy, just sort of a static turnover of small population. We heard in the disruptive and the stabilizing, those two types of sort of pathways that were described for how you might have persister cells in microbes. Some of it might be simply imposed. The cell cannot divide. It's enforced non-cell division, or maybe it's an adaptive response. It may involve no change in metabolism, but generally we think of it as being a reduced metabolism, not feeding, and then a reactivation cost. So what I've kind of circled here in this region are sort of the features that I think sort of fit Jay's expectation of dormancy. And I'm now going to show you that cancer cells don't really, at least what we refer to as dormant cancer cells, don't really conform entirely to this. So what are the properties of what are now recognized as dormant cancer cells? They indeed are non-proliferative. They are in an arrested state. However, they tend to still be feeding. They're still uptaking nutrients. There's little evidence for reduced metabolic rate. In fact, in many of them, they'll have a hypermetabolism, not a hypometabolism. They do indeed represent a resistant life history state in response to lack of oxygen, pH, acidic conditions, and as we're going to see, in response to therapy. And it does look like they involve a reactivation cost. So what does this mean is dormancy is an adaptation in cancer cells? Bet hedging, absolutely. So just about everybody now, as they look for dormant cancer cells, find a small fraction of these cells in any population of cancer cells within a biopsy, within a tumor, and that presumably represents a bet hedging strategy. Avoiding overcrowding, perhaps. Some people have suggested that under very crowded conditions, some of the cancer cells will try to migrate away. If they get swept away, wind up at a metastatic site in a new tissue, that they'll go into a dormant state. Avoiding SIB-SIP competition, I won't go into it, but I suspect that's highly unlikely in the case of cancer, mostly because if they did go into a dormant state to help their SIBs, they would be just as much helping all the other cancer cells around them. Habitat selection in time, absolutely avoiding adverse conditions. It does seem to be a way of traveling or hunkering down when things are bad and waiting for things to get better. Now, if you look at this metabolic continuum, I really want to put cancer cells in perspective. So we've already seen over here these deep dormant endospores, or any of these spores that were so beautifully described in Pilar's talk, where the metabolic rate becomes just a tiny fraction of what they otherwise could be. And then, of course, there's active states, hummingbirds, things like that. Across this metabolic continuum, where it's really very broad for microbes when they're dormant, actually the range of relative metabolic rate probably varies little between what we might describe as dormant cells and active cells. And as I said, it may even be reversed, where the dormant cells are actually metabolically more active. So what are we learning about what constitutes a dormant cancer cell? Is it just G2M arrest, or is there something more to it? What has emerged? Probably in the last 10 years, but certainly it has accelerated in the last 5 years, is a pretty amazing set of observation. And this is the role for what has been observed as giant cancer cells. Now, giant cancer cells were observed a long time ago. They were originally thought to be irrelevant. They occurred at a small frequency. They were thought to be senescent. They were thought to be abnormal in some way, and that all they could do was simply wait to die. Then people noticed there were these polyploid, that these giant cancer cells were polyploid. They had different properties. And it was finally suggested, starting this millennia, that maybe they play a role. And then the team at Johns Hopkins, with others, has been describing these polyaneuploid cancer cells, PACs, that may allow for essentially cancer cells to exhibit the adaptations of what we expect dormancy to serve in terms of the ecology. And then finally, persister cells, we've heard mentioned from the microbial world. Persister cells is also mentioned, and we're seeing a convergence of ideas. So we're going to take this crazy idea. This is this team from Johns Hopkins, folks from Michigan, Princeton, have had the pleasure to work with these wonderful scientists. And what was observed is if you stress cancer cells, they started with prostate cancer cells, but they've done this now with a large number of them, you stress them, and what happens is you go from an untreated condition, if you just look at DNA content, standard diploid cells, these could be cells in the G, these guys are in the G2M phase, so they have a doubling. And then you have this small group out here that has much bigger. They basically represent kind of polyploids. And what they showed is as soon as you face a stressor, lack of food, a drug, hypoxia, lack of oxygen, you see this whole shift towards these polyploid cells. They're beyond G2M arrest, They've proposed what's going on, and they've been doing incredible work, as other labs have been, to show that in response to the cell, in response to the stress, you're in sort of a G2M state. In that G2M state, perhaps at that point, you simply go on, you try to survive, you eventually die from the stressor, or you don't actually manage to get into this polyploid state and you go on and die. But the sum of them, through a process of endocycling, double, quadruple, even 8-fold, they've seen 16-fold increases in the DNA And then what happens is over time, these PACs, polyaneuploid cancer cells, then begin to depolyploid. So they don't breed true. They don't actually divide and maintain that level of ploidy, as you might expect for standard polyploid organisms. In fact, they depolyploid. They almost bud back into a 2N state. So what do polyploid cancer cells do? Well, obviously they're non-proliferative until they actually depolyploid. So let's imagine how they might be useful for solving problems, evolving solutions. So imagine an unperturbed population. This is sort of this water line here, is that if fitness gets below this line, the population is unsustainable. If you're above this water line, the population is sustainable. Note over here, we have the trait value of the cancer cells. This is sort of their fitness. Now imagine you have a catastrophic event. And at this point, the trait value is actually underwater. This could be in response to therapy, where if this trait persists and does not evolve, the cancer will be eliminated. So what are some of the ways by which cancer cells solve this problem? One is there can actually be existing genetic variability. It may not just be a dot. There may be a whole spread of heterogeneity, genetic heterogeneity. So there already may be individuals in the population that are at the waterline that can now evolve resistance. Furthermore, this population may go quiescent, dormant, and simply wait for the catastrophe to end, get back to the normal circumstances. The other is standard evolution, evolutionary triage, where you generate heritable variation and slowly climb your way up to the population. This is known as evolutionary rescue. Or here's a wild idea, self-genetic modification or self-epigenetic modification, where some of these cells are able to determine that they're underwater, that they are not sustainable, go into a dormant state or a state non-proliferative state, and then solve the problem. And then upon solving the problem, and all of you, I'm sure, thinking about, well, how can they solve the problem? Actually then begin to depolyploid with the solution. So what do we actually see? So it was then determined that, and tests done on these PACs, was maybe the PACs actually already have the mutation and they proliferate, hunker down until times get better. Maybe the PACs produce a blast of high genetic heritability, high phenotypic variability, and somewhere in this blast of offspring is an offspring that works, standard moving up the landscape, or this notion of sort of a self-genetic modification in which they actually are able to solve the problem without creative destruction, without births and deaths. So enter Ozma El-Kanawi, who's now at the medical center at Indiana University, and Bob Gatenby, head of radiology at the Moffitt Cancer Center. They studied these persister cells. And what they did was a fairly standard experiment. I had the pleasure of participating in these experiments. They would take lung cancer cells at about 20,000 cells per well. These were seeded. So each of these wells, about 20,000 cancer cells, they were all dumped into these wells. So you have a lot of multiple populations. They were then, subjected to a chemotherapy, erlotinib, which basically punches holes in the membrane. It actually ruins the osmotic capacity of the cell membrane, causing pyroptosis, which is basically a flooding in of the outside and then cell death. And several things were noted. If you added the erlotinib, in almost all cases, the populations crashed. In a few cases, shown here in blue, in a very small number of cases, there was already the genetic variability for resistance. So these populations would crash, and in those three little blue dots, the population would rebound instantly. And you would see an explosion then of resistant cancer cells, kind of the standard way by which resistance is thought to evolve. However, in the remaining wells, those are the red wells, there would be maybe 100 surviving cells. So this out of 20,000, there might be 100 cells spread out haphazardly across the well, and they were surviving. These cells were surviving, but they weren't doing anything. They fit the classic cancer perception of persister cell. But then the remarkable thing is sometimes as soon as 40 days, but usually around 60 days, which for a cancer cell is generation upon generation of generation, at about 2 months, they would begin to proliferate, and they were resistant. And you'd see these resistant morphs emerge. So what happened? Well, first, they're really different. These persisters lie in a very different space in terms of their phenotypes. They tend to upregulate methylation, meaning they appear to be exploring epigenetically the whole human genome. They also seem to have amazing danger sensing machinery. And then the truly remarkable observation is when they broke out, they were already fully resistant. They had solved the problem. And it looked like they solved it epigenetically. So here's their problem. They have an osmotic crisis, cell death. These guys, persister cells, appear to tolerate the osmotic crisis. They persist, and then they proliferate. And they're now resistant, and their offspring go on to continue to be resistant. And then upon observation, very satisfyingly, but not surprisingly, given the story I told you out of the Johns Hopkins team, if you look at them, these persister cells don't look like your ordinary cells. They're packs. They are these polyaneuploid cancer cells. They are these giant cancer cells. You can see it in the original group that spreads out of the original cancer cells down here. This is your standard group, a little bit of G2M arrest. And notice you get a big jump up here into these polyploid cells. So what does it mean? This is right now an exciting field. There's a whole bunch of labs that are exploring this. These packs seem to be a stress-tolerant state. They seem to form by endocycling, not G2M arrest. They're non-proliferative, but they are hyper-metabolic. In fact, lipid metabolism goes way up. There does seem to be this intriguing possibility that has been found in yeast and other systems of a self-epigenetic modification to solve the problem, and then they depolyploid. And these may be cancer cells that contribute disproportionately to therapy resistance and to metastatic potential. So, can we target them? And that's current sort of state of research right now. You exert chemo, you get these persister cells, these polyploid cells, and then you get the recurrence of the disease through resistance. Can you somehow target? Can you target the packs themselves? And right now, these are the proposed approaches. These are now being done certainly in petri dishes, beginning to see them in mouse models. One is, can you actually use blockers that prevent this asymmetric mitosis that packs require to depolyploid? So maybe if you leave them permanently in the pack state, as Jay pointed out, if you can never return from that state, then you're a dead end. Can you prevent the pack formation itself? Can you actually prevent them from entering endocycling? Are there ways to actually target the new metabolism, the novel metabolism of packs that seems to see a high degree of lipid use, target the metabolism? And finally, can you block the self-genetic modification? And this would be, for instance, through hypomethylating agents. So at the end of the day, what do I get to do? I'm at the Moffitt Cancer Center, Integrated Mathematical Oncology. We think a lot of this is interesting. Dormancy and understanding of dormancy from nature provides amazing insights into convergent evolution. It may help inform the kind of evolution that was being discussed, and it may provide new ways of treating therapy resistance. Ashley, thank you so much. I think I've gone way too long.
No, you're just ending right on time. Thank you so much.
Thanks.
And I'd like to just move into our discussion. For those of you participants that would like to stay a little bit longer, we got started about 8 minutes late, so we'll continue the discussion as long as as long as it's interesting to you into that time. We've got some great questions in the Q&A, and I'm going to apologize up front because we're not going to get a chance to get to all of them. But I think if the panelists could please turn on their videos. I'm going to start us off with the question about defining dormancy and the different concepts of dormancy that we've seen expressed today. I was making a list through all the presentations. It seems like everyone is in agreement that there's no reproduction during a dormant state. That seemed to be a commonality, that the dormant state is protective or recalcitrant to stress, for example, and that there's a cost. There's a cost for the reactivation property. Are we all in agreement about those three traits of dormancy? But then there's some other interrupt me if you have a good thought, but there's some other conceptions here about like whether we define dormancy simply by arrested growth and reproduction, or if we also define it by a reduced state of activity. In the case of the cancer cells, that's not necessarily true, but also I think in Natalie's talk, you were making a definition of dormancy based on, are you able to have an organized cellular response or not? And so the cells that were persisters in the cases of the GASP response, they were still, they were not growing, so they were considered to be persisters, but they weren't necessarily a low metabolic activity. And so I'd like to kind of dig into this concept. Should we have a binary definition of dormancy where you're either growing or not, and who cares about your rate of metabolic activity, or is that level of metabolic activity important for defining a continuum or a gradient? And who would like to comment on that first?
So I can, because I think in my talk I showed these two growth arrests. So if you just look at the cell, they're not growing in the same way, right? But one of them is metabolically very active, and one of them is clearly going into a regulated state. This CASP state is actually very regulated. They are smaller. They chew on their RNA, so they take down their RNA, they take down their metabolism, orders of magnitude compared to exponentially growing state. So there, I think we all agree that this is dormancy, right? Now, are we willing to call dormant cells just because they're not growing? I think it's just a matter of semantics. So, I think the field has to agree on whether dormant mean non-growing and that's it, or does dormant mean something deeper than non-growing? In my definition, I would call dormant only these regulated modes of growth arrest that actually take down the metabolism compared to the exponentially growing cells. But I'm open to suggestions if someone wants to define it differently.
Yeah, if I can just jump in, and then I would love to hear what Jay and Pilar think, because I think you kind of hit it actually. To me, particularly from my experience now being in the cancer world, to simply define dormancy as being non-reproductive, well, then I'm dormant. So clearly, that's probably many people would say, yes, you are dormant. So I think simply non-proliferative is not a satisfying answer. So I think, and this is in Jay in particular, and my awesome colleagues might be able to comment, I think it requires a state. It's got to be a definable state that is distinct from, say, the standard proliferative state or reproductive states, or potentially reproductive states of the organism. There has to be perhaps a cost of entry in exiting the state, but that this state serve a very specialized service in terms of adaptation. So I don't think you can, so in other words, I prefer a definition of dormancy that involves a little bit of the evolutionary purpose or the evolutionary function of this state may help us get out of this conundrum of whether you can be hypermetabolic, hypometabolic, not feeding, or feeding.
Pilar, Jay, any response?
Go ahead, Pilar.
No, right.
Look like I saw your microphone going on and off. I didn't know how to interpret that.
I think it's really, for me, it's really about these trade-offs. And I agree that we need to keep this into an evolutionary framework and what is the cost and advantages of these strategies. And also the fact that it has to be a temporary state. And that's the reason you need to define a state. Because as you mentioned, okay, you are not reproductive now, but who knows? You see, is you can change these states. And this is something that is key, being able to change. I think it's something, as Jay mentioned it at the very beginning, this resuscitation, I think we tend to ignore that part. And I think it's very key, because just being dormant and yeah, if you cannot get out of it, then you're dead. Functionally, you're dead. It's a very different state.
Jay?
I guess in the criteria that I laid out, I mentioned reproduction, but I guess more specifically, we could think about whether or not an individual is undergoing processes related to biosynthesis and growth. So even though Joel's not reproducing, his cells are undergoing mitosis. They are growing, and so therefore Joel's not dormant by my standard. But if you had an individual that all of a sudden was to shut down biosynthesis, it wouldn't still need to be completely inert. There are very few examples of biological cells or organisms that are completely metabolically inactive. There's always some maintenance. Organisms need to maintain homeostasis. They have to maintain energized membranes. They need to repair damage to their and eliminate damaged cargo in their cells. And so all of that is a requirement. Otherwise, cells will die. So the only thing I'll add to this conversation, I guess, would be is that there's no completely off switch to metabolism. There's always going to be some reduced, small, residual. The degree to which cells or individuals do that is going to vary. But in my perspective, so the idea of there being increases in biosynthesis and growth and calling that dormancy, I have a hard time reconciling that with the way I think about dormancy. But yeah, so I think there has to be some reduction in metabolism. That seems central to me, but I'm open to hearing what other people think.
I love this conversation. Thank you, everyone. So it seems like there's still some room to think about dormancy as far as whether it should be defined as a particular state or along a continuum. And there are some key features that we agree upon here, but there are some other features that maybe we don't. So I think that's interesting. I think also there are some interesting questions in the chat that came up because we mostly today talked about responsive dormant states. So a cell is stressed, it's responding to a trigger in its environment, and it's entering an inactive state. And so there were many questions about, well, what are these stressors? Can we define dormancy as a response to a particular set of universally generalizable stressors to cells? The example of temperature came up, for example, in the chat. Like if there's a sudden change in temperature, a cell's response to that, a sudden and presumably certain level of intensity of change. And so in Natalie's talk, there was some evidence that the duration of the stressor influences the dormant response, and in Joel's talk as well. And in Pilar's talk, you had this really interesting example of an extreme environment, if you will, extreme to the maybe to the humans, not to the microbes, of this extreme environment where you saw a complete overlap in what you might call the resistant or the lysis resistant community with the total community, suggesting that, the stress tolerance in that system, in that community is equivalent. There's essentially no distinction between the dormant pool and the total pool. So what I'm getting at here is there a way, should we define dormancy or define responses to dormancy based on common stressors or common responses, biological responses to changes in the environment? Is that useful, or is that just going in the wrong direction?
I think the problem for me is that, as you just mentioned, extremacy or these changes are very unique to the organism. So that's the reason it's very difficult to find a common extremity as a point of view. So as these examples from these deep geothermal reservoirs, we're talking about 150 degrees, 6 bars, and a lot of metals, radioactive elements, so just name it. And these organisms, that's their everyday life. So for them, being exposed to oxygen is highly toxic. So you see, I think indeed it can be a response to stress, but the problem is you will need to define these stressors almost individually and based on the somehow the natural conditions in which these organisms may be able to grow. At least that's what we have seen in environmental samples.
Any other comments?
Well, I think what's interesting is you're kind of getting into, I think, what's important in the distinction between sort of an obligate versus a facultative dormancy. So if you're bet hedging and then things may be great. In fact, things may continue to be great, but it's the evolutionary history that sometimes things go really bad. So in that sense, you often have, like in the case of these cancer cells, like in the case of seed banks in the desert of annual plants, you'll have a form of dormancy then that is this kind of obligate. And then we've also been describing this facultative. A stressor hits, I have time to assess it, shift into a more resistant state, or I'm able to anticipate that times are good again, so I can kind of reemerge from it. And of course, this facultative dormancy can either be seasonal, like in the case of the Arctic ground squirrels that I've worked with, and those guys, of course, it's a very predictable opportunity, or it can be very unpredictable in the case of the rainfall in the desert.
I mean, one idea is that maybe some low level of dormancy is physiologically or genomically, genetically inevitable. If I take a population of E. coli and grow it in LB at 37 degrees on a shaker under the most optimal conditions that we could conceive, and we were to look at the distribution of metabolic activity among all those individuals, they're not all going to be equally active. For random stochastic reasons related to gene expression, there's going to be some probability that individuals are not going to be as active as other ones. So this is perhaps one example of where random stochasticity in gene expression, I'm certain Natalie knows a lot more about this topic, could be important and can be acted upon by different environmental conditions of different populations, whether it's in a host or in the environment.
Natalie, do you want to add?
Yeah, so clearly there is a distribution of metabolic activity in a growing bacterial sample. I do agree. But I think what we do mean, I think, by dormancy is something that goes even below that, right? Something that would be clearly distinguishable than exponentially growing cells. For example, at the level of how much heat does a bacterium produce when it's growing exponentially, it's really orders of magnitude more than when they are in this growth arrested, nice regulated state, right? Mm-hmm. So when there is a big difference, I think it's very easy to say that this is dormancy, and I do agree with the fact that it depends on the organism you're looking at. For example, neurons, right? Neurons do not divide, right? So, but we don't call them dormant because, you know, if they spike, if the purpose of a neuron and the evolutionary purpose is always behind the back of our mind. If the evolutionary purpose of a neuron is to spike, then even if a neuron doesn't divide, when it's spiking, it's certainly not dormant. We won't call it dormant. So I think it is, dormancy would be a metabolic activity that is much lower than the typical metabolic activity of that organism in the normal environment, right?
I think this can be a stumbling block in trying to come up with definitions and conceptualization. Like, how inactive do you need to be? You know, the one question I received recently was like, well, when we as humans go to sleep, is this dormant? And I had thought about that question before, and I was like, I need to go and look it up again. And you can find people who study human physiology who will say that dormancy does match the definition of what sleep is, and other people who say it's not. And so we'll probably, you know, and I saw in the chat, somebody's like, well, should we be using the terms quiescent instead? And I feel that this language is kind of, in some cases, well, we're biologists, all of us here. So we are really intrigued by all the differences, the nuance, whether it's cancer cells or squirrels or plant seeds or your group of microbes or my group of microbes. But there's a common theme, right? And that is that organisms have the ability to exhibit different degrees of metabolism. And that corresponds with changes in protection and persistence. So I'm happy to, I've been accused of overlumping things and oversimplifying things, but I think in some ways we need to acknowledge the differences, but we also, if we want to advance things across disciplines, we also need a conceptual framework that brings everything together.
So then in that case, perhaps what we should be looking for to define our dormant states is this kind of punctuated change towards a low or non-reproductive state, what was the word you used, Jay?
Well, energy is used for making biosynthesis in the production.
Biosynthesis.
Anabolism versus catabolism would be another dichotomy.
Right. But there's still some basic maintenance, as you said, that needs to happen or else the cell is dead. But at the same time, you could be a cell that's non-reproductive and still contributing respiration, right? Still maintaining the repairing the DNA, repairing the cell wall, et cetera, maintaining itself as a living cell. And so should we be looking for all of these convergent evolutionary events in which all the different microbes and all the different eukaryotes have made a dormant state for themselves? Should we be looking for essentially a jump? And that jump may be different for Jay's favorite microbe than for my favorite microbe. But when I see a jump downwards, that's suggestive of a change in state, and that state is dormancy. And my state of dormancy for my favorite microbe is different than Pilar's state of dormancy for her favorite microbe. Unless our favorite microbes are the same, which, they might be. Are we looking at a dynamic? Are we looking at a jump?
So Ashley, I think I'm going to push back a little bit on this, or at least invite returning to an evolutionary framework. So when Jay throws out sleeping, for me to determine whether that's dormancy or not dormancy is what is its function? If sleeping is a way to regenerate sort of memories and clear the brain and serves a function and is not so much a state, just like sort of eating or any other kind of activity, I would not call it dormancy. But if sleeping, either by choosing the right location to sleep or the sleeping itself results in a resistant state to some of the stressors and challenges, that if sleeping is a bed hedging adaptation, which at least for humans, that wouldn't be satisfied. Well, Jay, that would be my criterion for when did he, so rather than going down into the weeds on is sleeping dormancy or not dormancy, I would fall back and invite the team to think about it within an evolutionary framework, because Pilar stated so clearly.
Well, it sounds like you could make an argument that sleep could be a form of dormancy depending on how you interpret its purpose, or its function rather. Yeah.
Yes, so I would invite us, can we have a definition of dormancy without its function? That's well put.
We have a definition of dormancy without its function. Can we know when cells are not dormant there's a question in the chat about a threshold for reactivation or an energy jump, which is kind of the inverse of what we were just talking about. I was talking about an energy jump down, and there's someone in the chat asking about an energy jump forward during a reactivation. So a cell has been dormant because all of a sudden you have an increase in metabolic activity, potentially leading to reproduction or increased biosynthesis. And so maybe the challenge is defining the state of activity rather than the state of dormancy, and being able to deduce from the state of activity if you've come from a dormant state. I don't know if that made sense entirely.
I think, we had a similar problem with defining persistence, right? And we try to solve it because, some things, some parameters are on a continuous scale, right? For example, resistance, right? If you define resistant to antibiotic as a continuous scale, you have an MIC, which is high or low. There is no resistant bacteria. It's not a jump, okay?
Right.
But in dormancy, I do think that we understand dormancy as a state that has an evolutionary advantage, and therefore, as a state, the fact that we say the state, it means that it's not really a continuous scale, and if you do many things to a population of cells, they will all come around a state, right, which is not discretely different from the metabolically active one, right? So we do see some, for example, two potential wells, and then the ball would be moving from one to the other, being active and inactive, or growing and not growing, right? So I think it's a little bit like a differentiation program. The dormancy, this is why we call dormancy mostly for regulated growth arrest, because we think that you can be an actively growing cell, but you can differentiate through some pathway, which I agree is evolutionarily relevant, into a state which is dormant. And this is dormancy, right?
Yeah.
We had really focused a lot on this differentiation. What you mentioned for us is very key because, for instance, with fungi, I have to admit that to become crazy at a point. So there are many just very simple modifications that allow you to persist, and with a high metabolic rate. Is how much do you invest in modifying the structures or doing this differentiation process to the point in which you ensure to the best your survival by reducing your activity. And in a certain way, that's reason for me it's always a bit of a problem with the spores and so on, because that's like the maximum way it can change as a bacterium when you engage in endosporulation. In the case of firmicutes, you cannot go backwards. You engage in that and to end up producing the endospore. So we had always at least trying to look if is this associated to a massive change, morphological, or in your case, in the case of Natalie, physiological change, gene expression change that really leads you to this second strategy. And in fungi, to really see it, to see this continuum of some structures that are more resistant, but you have a change when you become a spore or when you become an sclerotia. That's entirely different.
Right.
Humans
So, oh, I'm sorry, go ahead. One last thought.
If you can really have something similar in human cells, in cancer cells, do you really see this massive change morphologically? I know very little.
Okay. Thank you so much for this panel discussion. I'm so sorry we don't have more time to keep going, but I'm getting news that we should wrap up the webinar right now. Thank you so much to all the participants, to our panelists, and to ASM staff. We're looking forward to part 2 on November 7th, so make sure you register for that webinar if you're interested. And we're going to be talking about how we discriminate different states specifically of microbial activity and different tools that different researchers who are experts use to do that. And so it should be really fun, a good continuation of this discussion. Thank you all. Have a wonderful rest of your day, and hope to continue this conversation onward.